Right, thank you, Scott. Welcome, everybody. So our first session today is titled Accelerating Barley Improvement. The intention of the session is to highlight advancements in tools and technologies that accelerate and support barley improvement. One note, we will keep speaker introductions pretty short throughout the day just because of the time constraints, as Scott said. But each of the speakers has a pretty extensive bio available on the website if you are curious to learn more about them, or just go talk to them. How about that? So our first speakers, as you might notice, are going to be joining us from Zoom. Dan Langford is the Business Development Manager, and Kelly Houston is the Deputy Director at the International Barley Hub in Dundee, Scotland, which also is serving as the host for the International Barley Genetics Symposium in October of 2027. And with that, I will turn it over to Dan and Kelly.
Fantastic. Yeah. Thanks, Max. Let's see if you can actually hear me.
Let's see. We can.
Is that?
Let's see. Can we hear you there?
You can't hear me? Hello, hello?
Yeah. See, Ashley, is there a speaker in here, or do we have to run it off this? Try talking again, Dan.
Talking.
Unless you take care of that.
We're just going to work on this just 1 minute. Try it one more time.
All right, here we go. Try again.
Yeah, just hold on one moment.
Try again.
We can hear you, Dan. For whatever reason, the audio in the room's not picking you up, and we thought we had this all set, so. If anybody's smarter than I am about this, feel free to holler. I don't have a speaker down here. It's up in my room, but I think they're both in my room. So I don't know what else you do have on your room. Okay.
I think when he's sharing, there is a sound button on the top right that he should activate, but it would be on his side when he starts sharing.
Right. Dan, can you try one more time?
No problem.
You're not hearing that, are you?
Is anyone hearing anything?
We had this all worked out. Try one more time.
I'm still here. Can you hear me?
How does that sound? Is it good enough? Even in the back, we're seeing thumbs up even in the back. So we're going to go with that, Dan. Thank you for your patience.
All right. I can yell louder if that's going to help, but maybe not. All right, so good to go?
Yes.
Okay, fantastic. Well, I was going to say it's great to be here, but it'd be much better to be there. Unfortunately, events overcame us. We weren't able to come in person, but I hope everyone has a fantastic conference. Thanks for the invitation to chat today, and I'm especially privileged being first up this morning. Hopefully, this is probably the least technical presentation you'll see over the day, but I can ease you into the agenda. Today I'm here with my colleague, Dr. Kelly Houston. We're going to talk about things, give you a bit of an update on the International Barley Hub initially, and then get into gene editing and some gene editing activities being undertaken here at the James Hutton Institute. So, as I said, I'll just talk about the International Barley Hub. You may recognize the name a little bit. Maybe you've heard it, but you may not be exactly clear what it's all about. You aren't alone. We haven't been really good at communicating it, but as the name suggests, it's always been designed to be an international hub of collaboration with our favorite cereal, barley. And it intended to be a community as well. We're now entering a phase where the IBH is changing shape. Most importantly, we're going to be clarifying our goals and formal structure over the next year. So I wanted to give you just a little bit of a quick update on this and maybe tease you a little bit with what we've got planned. So by way of just a quick background, many in the room will probably know Robbie Waugh and various other researchers from here at the James Hutton Institute. The concept of the International Barley Hub was developed well over a decade ago now, but it took a few years for us to secure some real funding, £62 million of investment to build ourselves a nice new shiny building. Well, it's new and shiny now. It took a while to get there as well. Construction, unfortunately, was waylaid by a little thing called a pandemic that came along in the early 2020s. But thankfully, some of the activity happened virtually. We've set up sort of a global webinar series. We're operating virtually. But now the new facility is complete. We've got to a point where we need to start delivering on the promises we made to secure that funding. And that is about achieving that goal of building an international hub for collaboration and community around barley. So that's the point we're at now. And as it says at the bottom, at the end of the day, it is all about achieving sustainable barley with that global scope in mind. Losing my voice already. So a major change is that we want to move away from the perception that this is just a James Hutton Institute initiative, and we're moving towards a partnership that's supported by a broad base of stakeholders. We hope this approach will provide new perspectives and lead to a more targeted investment in barley science. And the structure you see there on the screen is probably very familiar to anyone involved in different associations and organizations around the place. We have a few details planned that will make it quite different, especially with the sort of global reach and perspective there as well. So from an academic opportunity, a lot of the opportunities and programs that we'll be running are still in development. As I mentioned, we do really want to increase visibility across the globe of where funding might be achieved, where there may be needs and demands, and especially to avoid any kind of replication in research and efforts across the globe. So it's a watch this space for 2026. You'll probably see a lot more information coming from us over the coming months about how the International Barley Hub is going to take shape going forward. But I just wanted to give you a very brief overview on our plans and events just now. I'll throw in a couple of events there, and I did want to mention the Worldwide Distilled Spirits Conference being held in Edinburgh. Obviously, you're all enjoying amber right now in beautiful San Diego. We have a series of PhD students presenting at that conference solely on sort of some of the genetic research around barley. And if you do have the opportunity to come along to WDSC, it'd be fantastic to see you visit to the James Hutton Institute as part of the technical program there as well. Okay, so moving on to the topic of today, I mean, we're going to speak a little bit about gene editing, but there's been a lot of activity over the last few months around the European regulations behind gene editing. So I wanted to take a few moments to just provide a broad overview of that. It's maybe a little bit redundant for those coming from countries where this has been possible for a very long time, but maybe there's some detail here that might enhance the opportunity for collaboration. Okay, so by way of a very quick background here, there's lots of words on the screen, but the overall summary is the genetic modification rules and regulations in Europe were implemented as far back as 2021. And basically back then the focus was more on the process by which organisms are modified, regardless of the end result. And over that time and with the progression of technology, this is now moving towards more product-based approach. You could say this is more about looking at it with the end in mind or the result justifies the means to some extent. And this is looks like it's going to be a little bit fairer as these regulations develop. So it is a long time coming. The reason for this reform that's just happened over November, December last year is that, you know, a lot of these technologies, as we're all aware, probably didn't exist over this period. So even CRISPR is still quite new or has been developed since 2021, and the regulations definitely didn't allow for these new biotechnologies. Also, the threat of climate change and the need for more rapid ways of developing new varieties is driving the need to change these regulations. And recognizing that, you know, a lot of these gene-edited varieties are, you're effectively just accelerating the way conventional breeding might result in new varieties. The regulations should match that requirement. So focusing on the two streams of regulations, this is the European Union one. Sorry, I should have mentioned on the last slide. Back in 2001, of course, the UK was part of the European Union, so we inherited those regulations. But with Brexit occurring a few years ago, that all sort of fell by the wayside. So the UK has taken its own path, which somewhat aligns with the European Union. But right now we have two sets of regulations that are developing in parallel. Hopefully, ideally, they will converge, but only time will tell. So currently, the European Union, back in December, came to a provisional agreement for regulating gene-edited plants. They're categorized as New Genomic Techniques 1, NGT2 crops are generally things that you might traditionally consider as genetic modification. So they've separated these out so that they can be managed separately. Each country in the EU will basically verify whether a crop is NGT1 or not. From a labeling perspective, there is no labeling requirements, with the notable exception of seeds. Genetically modified, genetically edited seeds will need to be labeled. I believe this is a result of the organic lobbyists basically wanting to be able to ensure that they have a clarity and traceability around their supply chains. So you do need to label those seeds. Notable exclusion when it comes to traits, though, is the herbicide tolerance. A lot of the motivation around bringing these new regulations into play was based upon sustainability and being able to achieve some of the sustainability goals that European nations have laid out. And the idea that building in herbicide tolerance to these crops potentially increased the use of chemicals on fields was not really that appealing. So that is a notable exception that they've drawn out of this NGT1 classification. The other thing happening in both the UK and Europe is that all the information is being made public, including any patents tied to new varieties. So I'll speak about that a little bit more in a moment. Just a quick update on geography. You'll probably be aware the United Kingdom is made up of 4 nations, and sometimes they don't play well together. And this happens to be one of those examples. So while it says the UK's Precision Breeding Act came into force in 2023, it actually currently only applies to England. So anything you see about this is really England only, doesn't include Wales, Scotland, or Northern Ireland. Again, I'm sure that'll change over time. But just to be clear about that, that's how that currently functions. So again, we're using yet another new terminology, precision breeding. I kind of like it. I mean, I think it's an effort on the UK government's behalf to distinguish it from genetic modification, and yeah, to allay some of that public perception, make it a little bit more appealing to the general public as well. So the process is roughly the same as it was previously here in the UK. You still need approval for marketing of new varieties. There's just a new process in there. But if it's being used in food and feed, there's a further process it needs to go through to get approval. Very vague sort of comparison. DEFRA is kind of like the USDA in the US, and the Food Standards Agency is probably a lot like the FDA. So I'm whipping through this really quick because it's, just the aim was just to provide a high level overview, and as was mentioned, we're really low on time. We've got a lot of big data, a full day to get through. It's worth noting one aspect here as well from the European perspective is that they have focused in on a limit around the number of edits and the size of the edits that might be allowable under this new NGT1 classification. In the United Kingdom, there's no limit there at all. Again, it is based upon the product, and if it can be could have potentially been created using conventional breeding, then the number of changes, the size of those changes really don't matter. This is just one proposal that the EU is considering where the number of edits is defined more by the size of the plant genome itself, but time will tell how this plays out going forward. So for us, from a research position, it makes life a whole lot easier. I mean, we've been doing gene editing here at the Hutton Institute for well over a decade, but it's always been limited to very controlled environments. So now, thankfully, we can just through submitting a release notice to DEFRA, we can go out into the field and start putting gene-edited crops out in the field to do our trials. So really exciting development to be able to go and do that here. Although in England, but as time passes, we'll be able to do it here more locally in Scotland as well, but that's not a huge burden. So the ease of being able to do this is fantastic. I hesitate to say it's not even really permission. We really just need to let the government know that it's happening, which is pretty much the same as what we currently do with our more controlled environment studies. One other aspect I just thought I'd mention as well is, well, not the intellectual property side of things. You're always going to be able to patent things like you always have as it relates to gene editing. But the transparency, as I suggested before, is somewhat increased. So nothing about gene-edited crops can be confidential or kept secret in the UK. As soon as you start working on it, it needs to be made public. In the EU, it's a little bit different. Again, they are specifically tracking patents as such. There is a little bit of concern there about people using patents to limit market access. So, as soon as you start working on gene-edited crops in Europe, you need to disclose all the patents associated with it. Interestingly, there is another aspect called a voluntary licensing intention. So I look at it as a way of you being able to market or make people aware of your licensing intentions. So if you do hold a patent on some gene-edited technologies, then you can basically start letting people know that you've got that technology and you're willing to discuss licensing. And this is going to be monitored by the European Union. And if there's any corrective action that needs to be taken, then they'll absolutely do that. All right. Well, why am I telling you all this? I'd like to think there's going to be an increase in the amount of funding available for international collaborations around gene editing as a result of these new laws coming into place. The United Kingdom is, I think it was north of £20 million in 2025 was invested in gene editing projects, research R&D projects. Believe the European Union will likely follow suit and have some fairly significant funding opportunities coming, especially international collaborations. Hopefully they'll start increasing as well. So across the Atlantic, I'd expect some more opportunities coming there as well. I did a quick search to look at what might already be out there. And interestingly, there seems to be something coming from Canada, from the NRC. There's an engineering biology joint funding coming in spring 2026. Not many details available. It also seems the CRCC New Frontiers Research Fund international topic might have some funding available for collaborative funding around gene editing as well. Although it's a little bit difficult to identify anything from the US at the moment, but we'll see how that pans out. But ideally, with this sort of greater ability to share material, hopefully this opens up a few more pathways for collaboration. All right, so that's my quick overview. Hopefully there was something of value in there. It was very quick and very light on the changing regulations, but things are moving in a positive direction, and quite quickly. So fingers crossed that yields some results. I'm going to hand over to my colleague right now, Dr. Kelly Houston, who's going to get more into the technical details of the research we've been undertaking. Thank you very much.
Thank you, Dan, for that interruption. I'm just trying to share my screen. Here we go.
See your slides.
Okay, so as Dan indicated, we've been doing gene editing here for about 10 years, or a little more than a decade now, at the International Barley Hub and James Hutton Institute. And I'm going to give you a couple of examples that are focused on a particular trait, beta-glucan, that I thought would be of interest to this audience. However, we've obviously used gene editing to target multiple traits over the years. Yeah. So we've used gene editing to answer a number of different questions. Firstly, can we target multiple members of a gene family that we think are involved in the synthesis and quantity of beta-glucan found in the barley grain, and to identify which directly contribute to this trait? And second, can we generate more subtle effects by targeting the promoter of gene or genes that are responsible for this trait to modulate it more subtly? So, for these early experiments that we carried out using gene editing, we used quite a standard P-Bract vector that had been modified to include a barley codon optimized Cas9, linked up and driven by a maize ubiquitin promoter, and a guide RNA that was linked up to a separate promoter, this time a rice ubiquitin promoter, meaning that this construct would be expressed in all of the tissues around the plant. As many of you know, CRISPR-Cas9 works by having two discrete components. One is the guide RNA, which is designed to the region of the genome that you want to induce mutations in. And this is linked up to Cas9, which induces mutations and cuts in the DNA. We exploit the non-homologous end joining pathway, and this is the cell's natural repair mechanism, that when the DNA is cut by Cas9, the cell will repair the mutation induced. However, sometimes it doesn't, or it changes the sequence in this region. And these changes are the mutations that we study and try and understand the impact on the phenotype of the plants. So when we first started out doing this over a decade ago, we were designing our guide RNAs manually, although this quite quickly changed, and we started using this website CRISPR that you might be familiar with. What I really like about this website is that it includes several different versions of the barley genome, most importantly the golden promise genome, as this is the cultivar that is routinely transformed across the majority of groups using barley transformation globally. This website will predict the efficiency of the proposed guides, and it will also predict off-target effects and provide links to the gene models so you can validate this. And additionally, it actually provides restriction enzymes for each potential guide RNA sequence, which means that you can use restriction enzymes to carry out a simple digest followed by a comparison of band size relative to the wild type on a gel to score large numbers of potential mutants, making a really quick and efficient, cost-wise screen available to you. We use agrobacterium-mediated infiltration of immature embryos to transform plants with our constructs. And from start to grain from our T0 lines takes about 40 weeks. However, after 15 weeks, we will have a plantlet that you can sample tissue from for DNA extraction and for genotyping to identify which of these lines contain mutations that have been generated by the Cas9. Typically, we either use a simple restriction digest approach, like I described before, but we can also use an indel detection by amplicon analysis experiment as well. And what this does is amplifies the region targeted using fluorescently labeled primers, and then you run this on a capillary sequencer to compare the amplicon size relative to the wild type, and if the mutant is heterozygous, as in this case, or homozygous. And again, this is a much cheaper and more efficient way of screening large collections of potential mutants in Sanger sequencing. However, anything that we think might be a mutant, based on this result, we will carry out Sanger sequencing for. The authors of the paper that this came from claim that you can use this down to a resolution of indels of 1 base pair. We struggled at that resolution to detect differences from the wild type mutants, but we did manage to get this working for slightly larger indels of 2 to 3 base pairs. So it's really useful for us. So now on to more details about the questions we were trying to answer using gene editing. So Guillermo Garcia Jimenez did his PhD with us here at the James Hutton Institute a while ago. And we already knew that CSLF6 was a gene that contributed to the synthesis of beta-glucan, but we wanted to understand if other genes from this gene family, the cellulose synthase like F and H gene family, were also contributing to this trait as well. And so we selected 3 other genes based on their expression profiles, and we designed guide RNAs to the first exon of these genes, being careful to avoid the highly conserved regions indicated by the vertical orange lines, which indicate the amino acids that are characteristic of this gene family, and also other domains indicated by the vertical bars with the white and black hatching. And so for each one of these 4 genes, using a single guide designed to the first exon, we identified multiple different mutations in different lines. And for each of these genes, we identified lines that had a knockout or a frame shift that resulted in a premature stop codon further down in the gene. So looking at some stats from the T0, as you'd expect, we had a range of different mutation frequencies in the T0 from 4.7, which I consider to actually be quite low, up to 35.3% for CSF3. And at this stage, as to be expected, everything was in a heterozygous state. And again, typical of the construct that we were using, we had small indels occurring predominantly in our T0, except for one line. One of our CSLF9 lines had a large deletion of 39 base pairs. And so for the rest of this talk, I'm going to focus on CSLF6 and CSLF9, as when we quantify beta-glucan in the grain of the mutants of F3 and H, we didn't see any difference between them and wild type, and also there was no differences in the grain morphology either. But what you can see quite clearly if we focus on CSLF6 to start with, we have a homozygous mutant on the left of your screen and a heterozygous mutant to the right of that. And you can see that compared to wild type, these lines are developmentally delayed, and this appears to be occurring in a dose-dependent manner, with the homozygous mutant having a much stronger developmental delay to the heterozygous compared to the wild type. These plants are not only developmentally delayed, but they don't yield as much as your wild type. And you can see as well that grain morphology is also disrupted compared to wild type plants. And these plants have a knockout of CSLF6 present. The CSLF9 line highlighted here is homozygous for a knockout of this gene. And again, unlike the other lines we identified containing mutations in CSLF9, but like CSLF6 mutants I just described, there is a slight developmental delay compared to this line and wild type, and there is also a change in the grain morphology too. We saw that the 1,000 grain weight of the 2 lines, the CSLF6 knockout and the CSLF9 knockout, was significantly reduced compared to wild type, and for CSLF6, we could attribute this to an increase in grain length relative to width. And when we stain developing endosperms that are quite late through the developmental phase with beta-glucan antibodies, in the top row here, we can see that the CSLF9 mutants on the right-hand side of the screen are the same as the wild type, but the CSLF6 knockout has a again has this gene dose dependent level of antibody relative to the wild type. And the next row of images show calcifluor staining to give us an idea of the kind of shape and contours of the grain shape in these cross sections. And the following two stains, Lugol for starch and TB for cell wall distribution, allowed us to identify some potentially like quite fine scale differences at the structural level, which I'm not going to go into the details of here, but this was published back in 2020 as part of Guillermo's PhD. And then we quantified beta-glucan in the mature grain using a chemical assay, the Megazyme kits, which uses a series of digests, and then you're able to actually quantify the amount of beta-glucan present in the sample. And as you can see, perhaps again, unsurprisingly, mutants that had were homozygous for a knockout of CSLF6 had virtually no beta-glucan present. The lines that had this knockout present in a heterozygous state had a medium amount of beta-glucan present in this tissue. And interestingly, the CSLF9 knockout for in this collection of mutants didn't have a disrupted level of beta-glucan, but we found through subsequent analysis it actually had higher levels of arabinose and xylose in the grain relative to wild type. So we identified which one of the members of this gene family were responsible for beta-glucan in the barley grain, but by the process of knocking this gene out and then observing the phenotype in mature grain, we also saw along the way that doing this actually led to quite dramatic changes in the overall plant phenotype. So we started to wonder if it was possible to actually start to more subtly modify the amount of beta-glucan in the grain, but without jeopardizing the rest of the plant's phenotype. So as part of Guillermo's postdoc, which was carried out with Monica Doblin and Tony Basique at La Trobe University, funded by a DP project by the Australian government, he started to target promoters of CSLF6 with gene editing. So Guillermo designed 4 guide RNAs to this region of the CSLF6 promoter. And when we started to look at the distribution of the mutations that we observed in the T1, we could ratify this into multiple different haplotypes. And here I'm just showing you a small subset of haplotypes that we observed. And what you can see here is that when we look at the relative expression of CSLF6 based on the haplotype of the mutations induced by CRISPR in the promoter, you can see that actually CSLF6 expression is decreased in 2 of these haplotypes significantly compared to wild type. I've included some data on the expression of THX1 here as well. As we know from other crop species, that THX1 can bind to the second intron of CSLF6 and moderate F6 expression. However, THX1 expression here is stable, so we don't believe that this is what's causing the differences in CSLF6 expression observed in these CRISPR lines. And then, as I showed on the previous page, haplotype 6 and haplotype 5 have reduced levels of CSLF6 expression, and they also have reduced levels of mixed-linked beta-glucan in the mature grain. And this isn't due to any other structural changes that we're aware of. For example, the amount of starch present in the grain of these lines is no different to that of wild type. Often what you see is that when you have a reduction in beta-glucan, that can be because of an increased level of starch or vice versa. But here, this reduction of beta-glucan is happening independently of what's happening to the starch content of these grains. And then lastly, we were interested to see whether or not this decrease in beta-glucan led to a change in 1,000 grain weight of the grain. And we see that that does happen in terms of haplotype 5. It has a significantly reduced level, 1,000 grain weight compared to the wild type. However, haplotype 6 grain, which has this lower level of gene expression of F6, a lower level of mixed-link beta-glucan in the grain does not have a significantly different amount of 1,000 grain weight compared to wild type. Overall, these plants do not look significantly different to each other as well, and the yield is more or less equivalent to wild type. We want to grow these out in larger numbers in a future generation and characterize them more fully, but we think this is quite a promising result, that we've been able to modulate beta-glucan expression by targeting some binding sites in the promoter to change the grain beta-glucan content, but not impact the rest of the plant phenotype. So what are we doing in the future with regards to gene editing here at the IBH and the James Hutton Institute? Well, Dan's already mentioned that we have this large cohort of PhD studentships at the moment working on a range of different aspects of the barley supply chain, really focusing on sustainability and training the next generation of barley scientists, whether they enter academia or go into industry. And so this collaborative training partnership is funded jointly by industry and by UKRI, the UK government. And Shanzi, one of the PhD students who is into her third year now of her project, is focusing on expanding the range of barley gene editing tools. She's been doing this in a range of different ways and looking at a range of different aspects of gene editing and trying to develop new techniques and new tools for the community. So one thing Shanzi's looking at is whether we can delete larger genetic regions, and taking an approach using a nickase-based approach which generates larger edits and increases gene target accuracy. And this works because nickase induce single-stranded breaks which are less mutagenic and more specific than double-stranded breaks. And so she targeted the PDS gene using a pair of guides 4 kb apart from each other that can then potentially delete the entire gene. And we use photo bleaching, which is a nice clear readout of a successful mutation being induced in this way. And so Shanzi was able to delete the entire PDS gene. She validated this using sequencing. And in her T0, she had 5% of her plantlets completely bleached, as you can see here by this photo on the right-hand side of the screen. And a methods paper is currently in preparation. Shanzi's also focusing on another aspect of gene editing that I think is really important for the barley community and other cereal communities as well, trying to expand the range of different cultivars and genotypes that we can use to transform and carry out gene editing experiments. So to date, Shanzi's been focusing on 10 cultivars that are important for the industry. These have been selected through her industry sponsor, and she's transformed them and then monitored their survival. So the 2 cultivars that she has the most success with currently is Laureate and LG Diablo, 2 spring barley cultivars that are widely used in Europe. And when it gets down to it, she actually only sees 2.8% of the remaining embryos from these cultivars able to regenerate into a plantlet. So one of the clear bottlenecks is regeneration. And so based on conversations we've had with the team at JIC and their success of using the GRF for GIF1 constructs to increase regeneration efficiency in wheat varieties, we're taking this approach as well. This work is ongoing, but hopefully we'll have some really positive results to report soon. So please watch this space. So I'd like to acknowledge a large number of people that have been involved in this work over the years, particularly 2 excellent early and mid-career researchers, Guillermo Garcia-Jimenez, who's now at the University of Melbourne, and Shanzi Kumar, who's doing her PhD with us here. And I'd just like to take this opportunity as well to highlight, as was highlighted in the very kind introduction, that we are hosting the International Barley Genetics Symposium in 2027, which up until a couple of days ago sounded like a really long time away, but now it's definitely becoming real. So please go to ibgs15.com to register your interest. And if you have any specific queries that you think we might be able to help you with, please contact us at ibgs15@hutton.ac.uk. So with that, I'd like to finish and just say that we'll welcome any questions that you may have. Thank you. Okay, was there any questions?
Can you hear us, Kelly? Oh, this is fun.
Hi there, my name is Hannah Ullman. I'm Montana State University. I was just curious on the research that you were looking at beta-glucan in the grain, if you also included malt analysis in that assessment of the beta-glucan content for the final product. Although it is really interesting to assess beta-glucan in the grain itself, the end users of this are going to see the grain as malt. And there's a whole cacophony of changes that can take place during that malting process. And so ultimately, I'm more interested in the beta-glucan levels after the grain is malted.
Yeah, that's an excellent question. We didn't carry out any micromalting of our CRISPR lines to date, but as part of the investment that was led to the new buildings and so on that we have now on site, we do have the capacity to do micromalting, and that just makes it a lot easier to be able to micromalt gene edited lines. Obviously, that wouldn't have been something we'd have been able to ask industry to do for us previously. So we are definitely going to do that. We have also targeted some of the enzymes that hydrolyze beta glucan during malting as well. So we have mutant lines of those as well that we can test, and so we'll definitely be moving forward with that. But yeah, it's an excellent point. It's sort of it's not quite the full pipeline yet, if you see what I mean. So we'll definitely be doing that in the future as well. Thank you.
Hi, Kelly. This is Shengming from USDA. Nice work. Could you go back to the timeline of your transformation, the slides?
Sure.
I forgot the timeline when you first get the transgenic planet. What's the time when you get it? It's 12 to 10 to 16 weeks.
Let me just get my slides back up.
Still being shared. Just go to a demo.
Yeah, this one.
Yeah.
What's I cannot tell the number because of the colors. When you get the planet in the tube, what's the number there?
Oh, this planet here?
Yeah.
So that's about 15 weeks.
15
and so
weeks.
that's the point where we can
at that timeline, what's the
take tissue, depending on how, I mean, sometimes you get them and they look like this, sometimes they are, quite small, but I would say once you've got to this sort of 3-leaf stage, certainly before this point here, you can take some tissue for DNA extraction and then start to genetically characterize them. Does that answer your question?
Sorry, Kelly, the mic. The question is, what is the transformation efficiency?
The transformation efficiency in general, I mean,
Shayman, was that in general the transformation efficiency, or are you talking about
experiment? I'm just trying to find where we have the mutation frequency here. As you can see, that varies, but you're obviously talking about the step before the transformation efficiency. It really does vary depending on the transformation round, and now that we've started straying into looking at other cultivars as well, we get a really big range in transformation efficiency. To be honest, now that when we're looking at some of the cultivars that aren't typically transformed, so things that aren't golden promise, the transformation efficiency is incredibly low. We're talking 1 or 2%. But that's why we're trying to boost that by using these constructs that have been shown in wheat to be much better at promoting regeneration in those lines. So hopefully we'll be able to boost that in the near future, and then we'll be able to transform our favourite cultivars and actually even maybe move into other genotypes and ideally land races as well. So.
Yeah, I can repeat it.
Right now, it's just the CRISPR-mediated knockout. It seems why CRISPR-mediated cannot meet.
The question, Kelly, the question was, did you try CRISPR-mediated gene knock-ins, or was it only gene knockouts that were tested?
I would have loved, I would have absolutely loved to have been doing gene knock-ins. And if you kind of go back to here, if you go back to one of the first slides where, it's sort of saying how we really are working in this pathway where we're relying on the cell's machinery to repair, but make a mistake. And then that mistake becomes the mutation that we study. Ideally, we would be looking at homology-directed repair here as well, where I think that's what you're asking about, where you provide the alternative version that you want the machinery to actually integrate into the DNA of your target. And unfortunately, we haven't got that working as yet. I think that I read previously that actually agrobacterium mediated transformation isn't the best for this. But it's also the homology-directed repair pathway is much less efficient, and the transformation efficiency of that is much lower than the NHEJ. But yeah, it would be something fantastic to get working in the future for sure.
I think we have time for 1 or 2 more questions. All right, if not, let's thank Dr. Kelly Houston and Dan Langford for joining us across the pond. And we'll move on to our next speaker. Thanks, Kelly.
Thank you.